is Dr. Yi-Liang Ding, who comes from the John Innes Center, UK, and she will deliver an interesting talk on RNA structure. Please, Yi-Liang.
Good morning, good afternoon, good evening. Thank you to the organizer to give us the opportunity to present our research program. So today I'm going to talk about RNA structure functionality in gene regulation in living cell. So we know that in the central dogma, in the textbook, you can see very complicated DNA drawing in the textbook. Also very complicated protein drawing in the textbook. However, when you see these messenger RNA in the middle, it's always drawn as a very simple line. So we don't know whether or not in living cell, these messenger RNA really looks like that simple structure. So over the past several decades, people put RNA with different lengths, different sequence content into test tube. It's always folded into certain RNA secondary structure. Sometimes you can see beautiful stem loop structure. And also it can fold into very complicated structures such as a pseudonode structure, triplex, also the G quadruplex. So then you're wondering if RNA structure can fold into different shape, why in the textbook these messenger RNA still draw as a very simple single line kind of shape? So then we actually noticed that all these study has been done under in vitro condition. So we don't know whether or not these structure will actually really fold in the living cells. And also, because we don't have that knowledge, we don't know the functional importance of RNA structure in biology content. So pretty much since 2014, several groups, including ours, developed the first in vivo RNA structure profile method, which actually take advantage of chemicals which can probing the RNA structure information inside the cell. So for example, dimer sulfate DMS, which can actually modify the single-stranded A and C and put a method group at their Watson-Crick phase. Because this method group attached to the RNA, so when you perform the reverse transcription, it will store at the position which the nucleotide got modified. So if you have more stalling at this nucleotide, that means this nucleotide will actually tend to be single-stranded. So by coupling with the Illumina sequencing platform, we generate a genome-wide RNA structure landscape. So based on how many reads stalling at a specific nucleotide, we generate something called chemical reactivity. So if you have a high chemical reactivity, that means you have more reads stalling at this nucleotide, then this nucleotide will tend to have a high probability to be single-stranded. So if you have a lower chemical reactivity, then that means you have less reads stalling at the nucleotide. That means the nucleotide will tend to be actually high probability to be double-stranded. So we also can actually slightly changing the reverse transcription reaction slightly to let the reverse transcription bypass these modified nucleotide by introduce the mutation. So you can also capture this by deep sequencing all these mutation to actually estimation the single-strandedness. So we also have another chemical which instead of modify on the nucleotide base, or we call it a shape chemical, which is actually can isolation the single-stranded RNA 2' hydroxyl group and put an RNA adduct on the RNA. So similarly, reverse transcription stalling or mutation can capture this kind of modification. Then we also can actually generate a shape chemical reactivity. So now with this kind of powerful in vivo RNA structure studying method, we thought, okay, maybe we can start to explore the functionality of RNA structure in living cell. So now we take a look at the textbook. We know RNA involved really important biological processes such as RNA processing, RNA degradation, and also translation. Again, if you're looking into all these textbooks, you can see these RNAs were all drawn in single lines. So do they really look like that in living cells? So now we can revisit the textbook by our powerful method. So the first question I'm going to demonstration just for a case study on the splicing. So the key question in the splicing is actually how does a spliceome recognize the true splice site? So we know GU and AG are splicing sequence motif. So however, if you go through the whole pre-mRNA, you can find many GU, many AG. So why some of them actually is the true splice site, but some are not? So we hypothesize RNA structure maybe can help a splicer to recognize the true splice site. So what we do is we generate a new method called the nuclear shape structure seek, which is we chemical treated the plant, then we isolate the nucleus, then enrich the pre-mRNA proportion, then generate a pre-mRNA RNA structure landscape. So this data allow us to ask the question how structure looks like to actually causing the splicing later. So based on that, what we find is actually 2 nucleotide single-strandedness upstream of 5 splice site is actually important for splicing and also alternative splicing. So when this 2 nucleotide is double-stranded, splicing won't recognize, so you won't have splicing. So if this 2 nucleotide is single-stranded, then splicing can recognize, then cleave the intron out. So based on this case study, one of discovery, we know that pre-mRNA do have a certain structure, and these structure features are very important for splicing. So now let's move to the microRNA mediated RNA degradation. So we know that this pathway is the major RNA degradation pathway in plant. So organelle protein will drive through microRNA to form base pairing with its target messenger RNA. So previously, people actually predicted a target RNA based on sequence complementarity, but we know that 70% of this kind of prediction is actually false positive, which means even you have a perfect complementarity between microRNA and its target, sometimes this messenger RNA won't go into the degradation. So then we're wondering whether or not the structure have a function or contribution to this biological process. So to do that, we develop a new method, which called a cap structure seek. So what we do is we chemical treated the plant, then we only enrich the intact RNA proportion, then generate the intact RNA structure library. So this intact RNA structure feature can allow us to know what the structure is actually help to actually doing the cleavage. So doing that, we actually find that the first thing we actually find is microRNA target site is actually folding into certain structure. It's not spatially available to the microRNA, which is quite understandable because if a 21 nucleotide is completely single-stranded, then you kind of thinking other kind of endonuclease will actually cleave it unspecifically. So the first thing we found is target site do fold into certain structure. It's not spatially available. So the other feature we find is a 2 nucleotide downstream of target site. If it's single-stranded, then organelle protein can actually cleave the messenger RNA out. So if it's double-stranded, then organelle protein couldn't cleave this RNA out. So we call this 2 nucleotide single-strandedness downstream of target site. We named that target adjacent motif, which we call TAM. So now we move to actually a very famous microRNA target example, APATO2, AP2. So AP2 is microRNA 172 microRNA target site. And we know that this messenger RNA is not cleaved by the microRNA 172. Interestingly, in our structure data, the 2 nucleotide downstream of microRNA 172 target site is form base pairing with upstream sequence. So then we wonder whether or not this is the reason why the AP2 could not actually cleaved by the microRNA 172. So since this target site is inside the coding region, so then we actually decided to mutate the base pairing partner, this A, which at the synonymous mutation site, without changing the codon to see whether or not we can change the fate of a microRNA mediated cleavage. So what we can see is if it's Y type, which actually this don't have TAM formation is from the base pairing, you see AP2 abundance is very high, so because the microRNA cleavage is not happening. So once you just changing this A to G to U to C, no matter what you're changing, the AP2 abundance dropping down, so because microRNA cleavage happens. So this actually really shocked us because single synonymous mutation can actually turn on the microRNA cleavage fate. So then we're wondering whether or not this kind of a structure change can really have a bigger genetic effect. So then we move to the plant. So what you can see is that in the Y type, we always have a very beautiful white petal. So in the AP2 mutant, you see these petal lost, okay? So when you complement the mutant with the Y-type sequence where the microRNA don't happening, so there's no TAM formation, no single-stranded base pairing downstream of the target site, then you see these white petal coming back, restored. However, when you have a synonymous mutation complemented to the mutant, because the TAM formation, the microRNA cleavage started, so you see kind of similar as defective mutant. So this actually gives us idea is you changing structure by changing microRNA cleavage fate really can have a strong genetic effect. So now we can update a little bit on the textbook on the microRNA mediated RNA degradation. So these target messenger RNA do have a certain structure which is important for this degradation pathway. So before I move to the translation, I would like to introduce a very famous RNA tertiary structure motif, RNA G-quadruplex. So what I do is using both Watson-Crick phase and also Hugenstein phase base pairing, you can form these kind of 4G forms a plane structure. So when you have like a 2-layer or 3 layers of this kind of plane structure, then you can form these kind of a beautiful RNA tertiary structure, G-quadruplex. So the sequence motif will actually have a 4 repeat of a G stretch, G sequence, also 3 Gs repeat 4 times. So this kind of a tower structure can be stabilized by potassium and destabilized by lithium. Over the past 30 years, people always don't know whether or not this structure really forming inside a living cell. So in my group, we developed a new method called Shape-seq, which is actually using the performance modification of NAR on the folded G-quadruplex to determine the folding status of G-quadruplex in living cell. So when the G-quadruplex is folded, the 2' hydroxyl group of the G of every last G stretch repeat, you can actually more expose outside. So this 2' hydroxyl group of this G will tend to preferably modified by NAI. So because this performance of NAR modification on the G-quadruplex folded status, we can actually estimation whether or not the G-quadruplex is folded in vivo. So by doing genome-wide actually analysis, we noticed that in the plant, both Arabidopsis and also the rice, we can find genome-widely most G-quadruplex is under the folding status. And in the rice, the folding status is much stronger than Arabidopsis. So interestingly, when we drop the plant into the cold for just 2 hours, we can see that globally, all these G-quadruplex get into much stronger folded status. So globally, a global enhancement of folding status in response to cold. So from the global analysis of molecular function on these identified in vivo RNA G-quadruplex, we find either this G-quadruplex can inhibit the translation or enhance RNA stability. So now I'm gonna just demonstrate individual G-quadruplex and show you how important of their function in plant growth and development. So one of the example we find is in this gene HERD11, there's a UTR region that contain a G-quadruplex we identified. So in this HERD11 knockout mutant, you see the root actually length is shorter. So when you actually complement with G-quadruplex structure with a G-quadruplex sequence back to the mutant, you can see this kind of phenotype restored like Y type, okay? Interestingly, when you mutated the G-quadruplex, just simply changing G to A, then you see the plant root getting longer. So then we later find actually this G-quadruplex on this HERD11 UTR region is actually inhibit the translation. So we know this G-quadruplex can affect the plant root growth through translation inhibition. So the other example we find is actually cold-induced G-quadruplex in a gene called COG1, which in its UTR region. So we know in the Y type, when you treat a plant with cold, the root will actually inhibition growing. So when in the COG1 knockout mutant, the whole plant is insensitive to cold treatment. So when you complement with the G-quadruplex, you restore this kind of a cold responsive growth. You can see the root getting shorter in the cold treatment. But when you complement with actually G-quadruplex mutant, you kind of lost this kind of a code-responsive growth inhibition on the primary root growth. So then we found that this G-quadruplex can actually enhance RNA stability. We know the G-quadruplex, this code-induced G-quadruplex in this COG1 UTR region can actually affect plant code-responsive growth through enhance of RNA stability. So of course, by reviewing the existence of RNA G-quadruplex globally, now we can update the textbook again, so we know, okay, G-quadruplex, this kind of structure feature can actually inhibit the translation, also the enhanced stability. So why all the research program in my group now is try to update more textbook, actually find a more important structure feature for the biological function. We actually want to push our technology to a completely new level. So we know that RNA structure is highly dynamic. So this is a one in silico prediction of RNA structure conformation for one single RNA sequence. So you can see it can form into different RNA structure conformation. So previously, when I talk about our in vivo RNA structure profile method, it's based on the Illumina sequencing. So which means we only can get the structure information for average ensemble structure conformation. So for individual gene, so we couldn't get individual molecular structure information. So whether or not we can push to get individual RNA structure conformation for single molecule in living cell. So I try to demonstration our new technology using a very famous plant long-non-coding RNA QLs. So FLC gene is a very important flowering repressor. So during vernalization, we know FLC expression will be actually suppressed after cold treatment. Then this repressor of expression of FLC is actually suppressed by its antisense long-non-coding RNA QLs. So QLs, because it's long-non-coding RNA, people actually suggested RNA structure should actually help QL to function as a suppressor of FLC. So, however, QL had a different isoform. So if using Illumina sequencing, we couldn't separate these isoforms. So then we developed a new method which not only can separate different isoforms for QL, but also we can capture the RNA structure information in living cells for every single molecule. We call this method single molecular based RNA structure sequencing. So we treat the plant again with chemical treatment. So we can obtain all the 4 isoforms of QL. Then I'll just focus on one isoform. Then we have all the RNA population that have a chemical modification, generate a mutation profile of single-strand cDNA, then generate a double-strand DNA. And this double-strand DNA later, we plug into the dumbbell DACT ligation into the PacBio system and perform the PacBio sequencing profile for every single molecule. So after that, we generate a high-field reads of every single mutation profile of every single molecule, then representative of every single RNA structure conformation. And then, of course, using this method, we can capture almost all the in vivo RNA structure conformation for this single isoform. Then based on the structure similarity, we can cluster these structures into different groups. So again, this major QL isoform, we can cluster into 3 different major RNA conformation clusters. So this is actually representative conformation. You can see one is about 85% of RNA population, 10% of this one, and also 5% of this one. So when we actually know QL is actually functioning in cold treatment, so we immediately treat the plant with cold, then we can see actually structure conformation change in response to cold in 2 ways. One way is actually two structure conformation actually have a proportion change. One is reduced percentage of a proportion of RNA. The other actually is increase of the proportion of the conformation. Interestingly, we also observe a brand new code-specific RNA structure conformation of a QL isoform, and actually it's coming up differently from the previous warm condition. So now we're wondering if we changing this kind of structure, how it's gonna happen. So we actually selected a region which is the most variable region across all the RNA conformations. So then we just basically disrupted the structure. So make it actually symbol like a bubble structure, very single-stranded. So then we got this mutated plant and value what's going on with the QAL function. We found that this mutated QAL is actually associated with the FLC DNA chromatin more than the Y type. Of course, this more association with FLC chromatin causing a big suppression on the FLC expression. Of course, actually give you early flowering phenotype. Okay, so today I show you all these new biological function of actually RNA structure we demonstrated from Arabidopsis. However, in the past couple of years, also in the coming years, we're gonna actually show more functionality of RNA structure crossing plant kingdom. And hopefully we can explore the evolution of RNA structure crossing plant kingdom, also actually find the new way to actually do the proper improvement. So I also want to say during the pandemic time, because accidentally we involved in the coronavirus project, we actually noticed we can using our technology to actually help to develop a new method for actually treat coronavirus. So then our institute in the past year established RNA structure guided design application platform we named as Definitely RNA, which actually is currently translating our new knowledge and also new technology to actually benefit a broader society. So we actually gonna start with 4 major applications with mRNA vaccine design, antiviral therapeutics, metabolite factory, and also molecular breeding. And we are really excited about the next phase of development. And I hope after my talk, everyone can think about RNA structure and can think about whether or not it will involved in your biological research program. Thank you so much. And I want to actually acknowledge bringing people in my group. They're actually really clever. They actually develop a new method. Also, I want to actually mention my great 2 mentors, Giles and also Caroline, who helped me develop these large scope of research program. Also my collaborators and also all the funding agencies. Thank you so much for your attention.
Thank you, Yiliang. Very interesting talk. And now is a Q&A session. Committee members, if you have questions, please feel free to open your microphone.
So hi, Yiliang. A very nice talk. And I was thinking about chloroplast RNA. The signal structure there has been shown to regulate gene expression. I mean, not really gene expression at the mRNA level or transcriptional level, but rather it's translational level. Using your technology, probably you can compare chloroplast RNA and nuclear encoded RNA.
Yes, definitely. So I think several groups which collaborated with us actually explored the chloroplast RNA. Also the in vitro probing showing these different chloroplast gene has actually very interesting structure motif in the 5-UTR regulation translation. Yeah, this is a part of actually we're doing with a collaboration in Germany. So working specifically on the chloroplast RNA structure regulation. Thank you.
Thanks.
Hi, Yiliang. Excellent talk. Yeah. So I'm wondering whether or not all the RNA is properly bound by RNA binding protein. Would you be able to deliver some kind of technology which can isolate the unique RNA structure, the same molecule but with different RNA structure and the probe, or try to isolate the RNA binding protein? Because we know so many RNA binding proteins, the function are not well known.
Yeah, I totally agree. So there's a couple of technology we actually is currently using. Also, we actually working on the RNA binding protein mutant. So one of the example is a glycine-rich protein GRP7. So we are working with the Doris Stagger to actually understanding how the RNA binding protein recognize structure and how they are actually regulation together. So the current stage we're using just a specific technology on the GRP7, but however, it will actually expand to other RNA binding protein as well. Thank you.
Okay, let me ask you another question or comment. Yeah, about this G quadruplast, right?
Yeah.
It was an interesting story. I think we have a couple more minutes there. When I was in grad school, Walter Gilbert's group first shown that structure happening in nature, in the test tube, right? It's so good to see that nowadays you can actually monitor that in vivo. That's very interesting. So in that context, I wonder, you said potassium, right? Potassium concentration is very important for the formation of that structure. Yeah, I'm working on potassium nutrition. I just wonder if low potassium would affect the formation of that structure.
Yeah, so potassium is the
Sure.
key. Also the sodium, also the calcium, which you possibly interested. So every these are actually can induce G quadruplex. So we do one by one actually in the lab now. So the salt treatment, also the nitrate, also calcium, which actually is the next phase of actually identify these kind of specific sensor. So we actually developed that on the way. It's just really interesting to see actually possibly there will be RNA sensor for these kind of different ion. So it will be actually really nice to see in the future work. Yeah. Thank you.
Oh, that would be great.
Hello, Ila.
Hi.
It's very nice talk. So I have a very quick question. Is it possible to predict the RNA structure, like AlphaFold, to predict the efficiently to predict the protein structure? If you can, because the same sequence, same RNA sequence have many different forms, but you can predict the most somewhat stable, the form, if you have the, enough of the data,
Yeah, I think this is pretty much everyone ask whether or not we can create a AlphaFold in the RNA. Yes, this is something which we really key into in the next phase. So current stage is we generate a different temperature with actually really neat thermostable. So we actually have every single degree change to actually towards the genome-wide structure. So we want to build up a very thermodynamic, super large database towards actually the next phase to predict the structure independent to any thermodynamic kind of parameter or algorithm. So which actually we cooperate with a couple of deep learning group. So actually try to see this is something which we really want to actually do the next phase. Yeah. Thank you.
Thank you.
Okay, Gideon, very nice talk. So I hope you know there's a viroid that can infect plants. So viroid is actually the single strand RNA, but they are very steep. So I'm wondering, so what factors affect the RNA structure stability?
Yeah,
You have any ideas?
Yeah, this is actually really interesting structure. So we actually currently work with in Wang's group on the virus specifically. Interestingly, we can actually find the G quadruplex also on this virus as well. So we think that there will be actually secondary structure shift between secondary structure and the G quadruplex to actually make it a function for the virus transport through different subcellular content. But at current stage, we still actually explore that. Yes, the virus is the most interesting structure in the plant system. We really actually key to actually get a better idea how structure functionality in the virus. Thank you.
Okay, thank you.